All right, good morning, everyone, and thank you for joining us for the Winship Grand Rounds this morning. If you are an Emory University or healthcare employee and would like to receive CME credit hour for attending today, the access code is 677522 and can be found in the chat feature at the bottom of your screen for our virtual attendees. If you have any issues with this webinar or the CME login, please send Janelle Taylor an email or drop a note via the chat feature. And this morning, we are very pleased to welcome Dr. Betty Y.S. Kim, MD, PhD. Dr. Kim is a practicing neurosurgeon specializing in brain tumor surgery at MD Anderson Cancer Center. Dr. Kim's laboratory takes a multidisciplinary approach to the research of solid tumors with a special focus on malignant primary and metastatic brain tumors. Her work has been published in journals such as the New England Journal of Medicine, Nature Nanotechnology, Nature Biomedical Engineering, Nature Reviews Immunology, and Nature Reviews Drug Discovery. So relatively unknown journals, clearly. Dr. Kim's lab is interested in understanding the molecular crosstalk that occurs between tumor and stromal cells within the tumor immune microenvironment, while also developing multiple patented therapeutic strategies to inhibit tumorigenesis. Her current research primarily focuses on finding new ways to promote immune recognition of glioma cells to boost the efficacies of cancer immunotherapies. So let's welcome Dr. Betty Kim today for her talk, Immune Engineering Approaches to Cancer Therapy. Thanks.
Well, thank you everybody for coming to the lecture. And thank you so much for the invitation, Kim. You know, you're getting old when your students who are sort of, you know, in their training are now like, you know, taking these like great positions and looking for jobs and things. So it's a really good feeling to sort of see what goes around comes around, right? So so what I wanted to do, I changed some of the slides this morning, actually. These are some of my conflicts. And what I wanted to do is just go over some sort of big, broad picture of what my laboratory focuses at MD Anderson. I'm a professor there with 80% research protected time, and 20% of my time we focus on primary and secondary brain tumors. But also, these are two sort of big stories, nanoengagers, which Dr. Kristen Huntoon was intimately involved in and had a publication that came out recently. A way that looks at why immunotherapy really doesn't work in solid tumors and finding sort of ways to improve that rate of response. And then also an area that we've been working on for the last 10 years or so, which is delivering genes at the mRNA level and utilizing a platform where it doesn't really interact with your body per se, and it can bypass a lot of the physiological barriers. I'm not going to go over this sort of slide other than that at MD Anderson, we try and go around the tumor as a way to take out as much of the tumor cells as possible, decompress, debulk. But also we feel that this perhaps allows us systemic therapies, if they're able to reach in the brain, to do less of its work in sort of different ways and adjuncts that we have, as you know, the awake craniotomies, where unlike any other areas in the body where you can take out a little bit more if the margins are going to get negative. In certain areas in the brain where it's quite sensitive, eloquent areas where it's flanked by speech or motor, every millimeter counts. So we have developed a method, probably here as well, where we can really get up to about 3 millimeters or so within the functional areas like motor and speech, and the patient, in majority of times, will be okay. So that takes just many, many years of sort of looking at patients in different ways. So here, I just wanted to show in terms of like, why is engineering all that important? I was a resident at the University of Toronto when I realized that the technologies and tools that we have for cancer biology was quite limited at the time, and wanted to maybe look at ways to develop devices, handheld devices, and multiplex sort of systems to allow us to detect, for example, CSF fluid, which was a problem for diagnosis at the time, where patients were staying a long time to get the beta-2 transferrin that was sent out every week or so in Toronto. And so I began sort of this journey as a PhD student in biomedical engineering And then you can see where residency training happened between this paper and the next paper. And so we continue to sort of work on this pathway. And so my lab currently works on these sort of 6 different areas that are intersecting. And what's really cool is that different students will come in with different aspects of these circles in their training. And what we try to do is that by the time they leave the lab, that whether if you're a chemist, hopefully you'll learn more about the in vivo and biological systems. If you're a bio person or immune person, hopefully you'll learn how to conjugate nanomaterials or conjugate different protein products together. And so that you know at least the concept of protein engineering. For the purposes of this talk, I wanted to talk about these 2 sort of elements highlighted here. This actually, Kristen, believe it or not, we did hire someone after 4 years of COVID and just Houston in general, difficulty in attracting high sort of technology folks. This is a multiphoton imaging. And what that is that you can take an animal, inject the tumor in the brain, since we're interested in the brain tumor, and we essentially do a craniectomy in these patients, the animal patients. We take out the bone flap, and on top of it, we actually place a glass cover slip over it. What happens is that these animals just, play around, and every time you want to image these animals after therapy or whatnot, you can actually image them in real time as they're anesthetized at the time. And so this here is a photo. I think the video stopped, but this is what happens to tumor vessels during vascular normalization strategies. So you know that blood vessels in the tumor are physiologically abnormal. They're leaky. They're just functionally abnormal, but they also look different as well, where they're very tortuous. The branch points are very, very erratic. Versus the normalish, normal blood vessels in the brain, they're quite streamlined, like great highway systems. So in this model here, we have tumor vessels. You see these sort of black spots. These are single red blood cells flowing. There are certain regions in the blood vessel that are stagnant right here, and some that have increased blood flow. And so if you imagine these green or cancer cells, that there's going to be really hardy cells, if you will, where they're going to grow even without oxygen or blood because they'll grow at anything. And so I think those are the cells probably that become dormant when all of the other cells that we're able to treat with chemo or immunotherapeutics are able to sort of die off, and then the tumors come back with a vengeance. And I think these are sort of the stem-like cells, if you will, or there's a gradation of how much these cells respond to oxygen and food, blood. But then if you normalize the vessels by giving them not a high dose of VEGF receptor, vascular endothelial growth receptors, Avastin for the mouse that we use here, but we give them a very low dose longitudinally, and around 8 to 10 days of this therapy, where your goal of giving this VEGF receptor, which we do in Avastin, where you sort of blast the brain, hoping that you'll kill off the blood vessel, hoping that will strangulate and starve the cancer cell to death, and we know that doesn't work. But in this model, what we do is we give them a much reduced dose, multiple dosing, and at a certain point in time, these tumor vessels actually straighten up. They become less tortuous, they actually become less leaky, and during that window period, you then shoot your therapy of interest, nanomaterials or antibodies, exosomes, what have you. And what we see, which I don't show here, is that the materials actually go beyond just the perivasculature if they're able to get there. And so engage with the cells within the tumor microenvironment. So by just changing the blood vascular system as a way, you can see that nanomaterials, rather than being clumped, this is the blood vessel and a distance away, majority of it within like 20 microns, you can actually, just by changing the vascular structure, you can actually improve the distance in which the materials can traverse. And what is also important is that because functionally now the vessels are different, where they're more oxygenated in the tumor microenvironment, your immune cells, such as microglia and peripheral sort of tumor-associated macrophages, they actually transition to a more inflammatory, or they respond to more of a pro-inflammatory state. These I won't go over, just to show you cool pictures. Anybody interested in imaging Alzheimer's plaque as they grow? This is ApoE positive sort of animals that we sort of image over time. A question of whether the plaques themselves are toxic. And that's why you would get these neurodegenerative effects, or is it that these are aftermath of a toxic event that happens and there's an accumulation of these plaques? And so this was just recently published. And this I thought was a cool picture where this is actually a single microglia. And a study that's not related to cancer, but I love showing this because we are continuing with this. And in fact, we found a collaborator who does a lot of this stuff. So something that's like new, Kristen doesn't know. She knows everything about our lab and more that I'm aware of. But this is a single microglia that we stimulate with a laser. We don't kill it, we just injure it just enough that they're unhappy. And what's really, really interesting is that you have all these other sort of microglia surrounding it that sort of sends these fingerlings around and wraps around this injured microglia. And what we're trying to dissect with our collaborators at University of Houston is, what is it that they're doing? Are they trying to wrap around this injured site and prevent inflammation from happening, or are they bringing in factors to sort of repair? And what's really interesting is that if you take a very young animal or an older animal, the rate in which these fingerlings protrude and retract are quite different. This is just looking at T cells flowing in the blood vessels. So these blue sort of cells are the vascular endothelium, and you have these red sort of T cells that are flowing, and depending on the tumor microenvironment, they'll sort of marginate. So here, there's inflammation that's going on beyond this corner, and you can see these T cells that should be flowing in the center, sort of marginating along the vessel and sort of leaving. This is just a calcium unchanging. The yellow parts are just calcium that's released at the astrocytic foot process, sort of the blood-brain barrier level. So what you see here is that you see a cross-section of a blood vessel. You have astrocytes that are wrapping around it. And when we try and stimulate the foot process with our laser, you see the release of calcium. That's calcium unchanging. So the reason why I'm showing this is that these are technologies that I think I would not have known about had I sort of focused on signal transduction and sort of the traditional ways of studying cancer, which is really important. But I think with the advent of new technologies now, looking at these models, this 2-photon at the time when we had developed or helped to develop it 8 to 10 years ago, these are prototypes that we're sort of just starting to use in animal models. And so I think these are just an example of how we can look at essentially cellular processes that are occurring in real time with new tools and technologies, which I think is really cool. There are a lot of challenges, when you're looking at, MD Anderson's cancer model and cancer. How do you do that? There's a lot of old challenges, and that is that GBM or glioblastoma and tumor in general are very heterogeneous. There are certain areas that are necrotic, certain areas that are hyperproliferating with vessels and cells and whatnot. And they also change over time as the journey of that cancer cell sort of become dormant and resistant. They mutate and change over the lifetime of that host. And so you need to change your therapy and sort of personalize it, if you will. And we haven't done that. We sort of, give one therapy fits all, and sort of we're stuck in that sort of mode. And I think that needs to change. For us, blood-brain barrier obviously is a major physiological barrier that we need to learn how to bypass, if at all. I think with immune cells that are able to now bypass without the need to sort of create drugs that will do that, it might not be such a big sort of hurdle anymore in the future, that is. There's also resistance mechanisms that are intrinsic to the tumor microenvironment that I think happens over time as the tumor microenvironment becomes more and more hypoxic and hostile, where the pH changes and the immune cells are no longer happy, and so they're not able to kill off these cells. And then you also have these toxicity issues. Whatever therapy you give the host, you have toxicity that's systemic or local, also immune toxicity that's a real major concern. So this, I think, is a messy slide, but a slide that I think sort of explains our entire lab in a nutshell. And that is that you have an invasion in your body, whether it be cancer cells, infection, what have you. The first responders to any disease is really your innate immune cells, and these are the macrophages, dendritic cells. And as you know, immune cells in general are sort of separated into two branches: the innate immune system, which is the first responders. They'll just respond to anything that's abnormal and go and clear it. And then you have the adaptive immune system, which is the T cells. These are the more educated ones, where they kind of wait until they're educated, and then they strategically go and kill off whatever they're educated to kill off. And so tumor cells that are growing typically should have these innate immune cells that should recognize it. And because these innate immune cells, such as macrophages, are also phagocytes. They actually go and gobble it up and eat it up. They actually then digest the tumor cells in the cytoplasm, and they present these little tumor peptides on the surface of these macrophages, which are also professional antigen presenting cells. And when that happens, T cells then recognize it. These are initially naive. They recognize it, they're educated, and then they're primed to then, in the future, whenever they see this tumor antigen, they go and attack it and kill it. A subset of these also transform into these memory T cells that allow them to sort of remember in the future, if they were ever to encounter this type of a tumor, to go and sort of kill that off. Unfortunately, there's a lot of sort of ways in which cancer cells can evade this whole process. One of the sort of markers that you probably all aware of already are the PD-L1 on the tumor side that are upregulated that prevents the phagocytosis from occurring. But there's also CD47, which is overexpressed in a lot of solid and liquid tumors. And that is that, whenever you have overexpression of CD47, phagocytes just don't engage in phagocytose cells. And so there's been clinical trials that have shown that antibodies against CD47 can block this don't eat me signal that allow more phagocytic sort of process to occur. And then you have the T cells that go in and kill it. So there's a balance between the don't eat me signals that are usually upregulated in tumor cells with the eat me signals. And these sort of boxes sort of delineate what markers we are actively studying in the laboratory now to either shift the balance towards more of the eat me pathway. So we know that phagocytosis in general is just regulated by different markers and different sort of ways in which different cells and tumor cells respond. In particular, the way in which liquid tumors, such as bloodborne tumors, leukemia, lymphomas, respond to these phagocytic checkpoint blockades, like CD47 blockade, is very, very different from how solid tumors respond. And so we wondered, is there a difference, why is it that the same cell from different origin, solid versus liquid, respond to a same antibody in a different way? Well, that story begins with the three really great postdocs in our laboratory, Kristen being one of them right here. A story in which we try and trick your body into thinking that a solid tumor actually is a liquid tumor and see whether the CD47 blockade as a proof of concept is effective. So the story begins with this marker, SLAMF7, self-ligand receptor for signaling lymphocytic activation molecule 7. This is a marker that's critical for allowing innate immune cells to detect. So it's overexpressed in a lot of liquid tumors. And the way it works is that SLAMF7 is also expressed in liquid tumors, but also in myeloid cells, innate immune cells. And it's only when you see SLAMF7 on the tumor side and the myeloid side, they interact and homodimerize. So you need to have two of the SLAMF7 working together, and that togetherness, if you will, increases the phagocytosis rate. What we saw is that in our study, and others have shown, that SLAMF7 actually is not expressed in solid tumors. And so what we did in this cartoon, as you can see here, is that we basically took a model where we wanted to essentially target our system. We don't want this phagocytosis to occur everywhere in the body, because then you'll have like a major autoimmune reaction and it'd be bad. But you have cancer cells that overexpress certain markers, such as HER2. So we took a HER2 overexpressing cancer cells, which is in breast cancer cells, for example. And we basically, what we did was we took a nanoparticle. It doesn't matter what type, but a spherical nanoparticle, and we conjugated the surface with two arms, if you will. We had an antibody that attaches itself to HER2. You can see that here. And so this nanoparticle will only bind to cells now that have overexpression of HER2. On the other end, we also placed SLAMF7 on it. So if you can imagine, on the surface of a HER2 overexpressing cancer cell, you have this nanomaterial that will specifically coat the surface of these HER2 cells, but on the other hand, it also expresses SLAMF7. And what happens is that whenever this sort of interactions occur, the myeloid cells will see SLAMF7, will think that it's a liquid tumor, and start engulfing it. And so this is just a series of results that we were able to sort of show that 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 tells us that this whole system works and according to our hypothesis. And with any nanoparticle based work, you have to really characterize the materials themselves. So we looked at their size through the dynamic light scattering. They're about 100 nanometers. And we saw that when we took HER2 overexpressing cancer cell lines like SKBR3, these are human cell lines, we can see that the BiTE, which is a bispecific tumor nanoengager, we just made that up, that it actually, we see that it binds to HER2 overexpressing cancer cells, as you can see SUMUP7 here, but we also see co-localization of the HER2 antibody, showing that, you know, that the bispecificness of engaging the nanoparticle to the HER2 positive cancer cells is present with the SUMUP7 that are now studied on the surface. We don't see that reaction in this MDA cell model because these are HER2 negative cell lines. So we looked at all the different types of binding constants. People asked, you know, what is a ratio in which you have to have your HER2 antibody to the SUMUP7? Is it 1-to-1 ratio? Do you need to have 5, 50, or whatnot? And it looks as though 3-to-1 ratio of SUMUP7 to HER2 antibody on the surface of these nanoparticles for this particular size, because that ratio changes depending on the size of these nanoparticles, is the best at inducing phagocytosis. So we use that ratio for all of our remaining studies. And this is just to show that when we take our BiTE therapy now and also treat it with CD47 in the context of a cold culture model, so you now have HER2 overexpressing cancer cells that are growing. We place macrophages in the cell culture system and place the nanoparticle with the CD47. You can see that there's a real increase in these black bars here of phagocytosis as compared to all of our other controls. And this is our positive control, where it's a leukemia cell line where these are liquid tumors. So with CD47 should just phagocytose. Here we, in the co-culture system, show that when you have cancer cells that are in the green, macrophages in the red, and you put them together, not a lot of phagocytosis that occur if the cell lines do not have HER2 overexpression, and you place the BiTE therapy. But in contrast, when you overexpress HER2, you see a lot more of these cancer cells being engulfed and shown demonstrated as being yellow within these red cells. This is just a quick and dirty assay using a particular antibody, the sinefical sort of antibody. What happens is that this antibody will only bind to an MHC, which is sort of expressed in myeloid cells that have a specific peptide. And here we sort of induce this chicken ovalbumin, which should not be present in a mammalian sort of model. And so when you have increased sort of detection of this antibody, it sort of indirectly tells you that antigen presentation has occurred in a sort of an increased format. And you see that here in the black bar. And so far what we have here is that the BiTE is specific for HER2 and is targeted. B, it increases phagocytosis, so they're ingesting more of these cancer cells. C, that now that they've ingested these cancer cells, they're actually presenting these tumor antigens at a much more efficient manner. And this is just to show that when you look at how T cells are now interacting. This is a messy slide, but I just wanted to highlight these 3 black bars, where if you have a finite number of T cells that are uneducated and they're just floating around, and now they're being primed and educated to detect a certain tumor antigen, you would think that the number of the naive T cells will decrease over time as you increase the number of memory T cells. And that's what you see here. You see progressive decrease in the naive cells as you increase the central memory and effector memory T cells. And what does that mean in vivo? And so we took this animal model, it's a syngeneic animal model, immune competent, and grew these breast cancers in the mammary fat pad. And we looked at their sort of tumor size over time with this therapy, where we sort of inject intratumorally the BiTE plus the CD47 blockade. And we see that these animals, we can sort of salvage about 60% of these animals for long-term survival. We take similar sort of animal system without the HER2 targeting, and in the 4T1 model, we see that there's really no efficacy. And so this tells us that you really need the HER2 as a initial sort of engagement and targeting system. We also show in this slide here that when you take these animals, the tubal animals that should sort of survive long term but deplete the CD8 T cells, that the effects are completely abrogated here. So that tells you that ultimately it's the CD8 T cells that are really important for maintaining long-term survival. When we take the blood of these animals, because remember, we injected the treatment intratumorally, but now if we take the blood of these animals, we show that there's an inflammatory cascade that occurs, that there's systemic sort of cytokines that are released that are pro-inflammatory, allowing your immune cells to work at a much efficient manner. And so what happens here is that we tested this in 4 to 5 different models for breast cancer. And there's always like 1 or 2 models that just really, they don't respond as well. I mean, you know, these are the poor responders, as we're calling them. Their survival curve is not as impressive. And so we wondered why. We did some analysis, and they took a couple of months to figure out. But we did some analysis on the tumor side, and we basically showed that the PD-L1 is upregulated in these animals that don't respond. And we also showed that when you give them the PD-L1 blockade, I should go back, that we're able to salvage about 40% of these animals that were essentially not responding to our BiTE therapy. So it's like a combination therapy, if you will. We also show in the study, just as a mechanism, because it's not just a show and tell, but in this study, we also look at how does this whole mechanism work, and it looks as though the innate immune sensing pathway, the cGAS-STING pathway, which you may have heard about, is an important pathway that sort of alters this entire process. So the editors then asked us, hey, you know, can you act, is this a, if this is a quote unquote platform, can you sort of show whether this system can be used for different types of cancers or different markers? And so we took a completely different model now, a triple negative breast cancer model that's widely metastatic and very aggressive, affects a lot of young women. We took these animals and what we did was we targeted not HER2, but folate receptor in these animals. And so we had 2 treatment arms. This treatment B is probably what happens to a lot of patients where they sort of show up to the emergency center or to the clinic with a breast lump. They get a biopsy or surgical removal of the lesion, and then they undergo this adjuvant therapy. And here we gave these animals PD-1 blockade, CD47 blockade, as well as our BiTE treatment. This is sort of the neoadjuvant group where perhaps they didn't, you know, we start off with, you know, a lump. Maybe they had a history of, you know, tumor, triple negative breast cancer in the past or whatnot. We intradumorally inject the BiTE therapy, we remove it, and sort of observe them over time. And regardless really of which neoadjuvant and adjuvant, we see the same effect in that we have good tumor control, and about half of these animals, about 40% or so, depending on how aggressive the model is, live long term. And you can see that the BiTE therapy really minimizes the degree of metastasis forming. You can see a lot of the animals dying of metastatic burden. And so we're continuing to sort of study this and looking at ways to perhaps not just look at SLAMF7, but other markers that allow phagocytosis to be engaged. And the issue that we're facing as a group, and something that, you know, I think depending on whether you're like a really pure nano, you know, pro nanomedicine person or not, I think it's, you know, the field will go in the middle. I don't think that nanomaterials right now in the current sort of era will save the day, and it hasn't, despite hundreds of billions of dollars that have been poured into the field. And in fact, the NIH is meeting in the fall, actually, to discuss how, like, what is the bottleneck and why have we failed despite having, you know, significant amount of investment in nanotechnology and nanomaterial design. And these are some, I think the 2 boxes really apply to the nanomaterial world, and that is the synthesis, depending on where and who synthesizes them, and what material, you know, was it metallic, is it polymer, what kind of polymer, what shape, what size, all of these things play really different sort of, they have a different role in that particular tumor and how tumor cells actually engage with these foreign materials. And then the other issue is that a lot of these are foreign materials, and so our body is really good at clearing them. And so the liver takes up majority of these materials. So that's another thing where, you can't really bypass the liver. The majority of them are not able to pass the reticular endothelial system, the liver and the spleen. But also the surface modification, you saw how important it is to target them, because without targeting, you don't want these sort of adverse reactions where normal cells are now being phagocytosed. You want it specifically for tumor cells, for tumor scientists. And so the way in which modify, and right now our lab is heavily dependent on antibodies because of the fact that these are readily available and certain cancer cells overexpress certain markers, but also peptides as well. I think the biological systems and animal systems that we have is applicable to all cancer researchers, in that, we can cure cancers in animal models, but unfortunately, when we translate that into the patient, we don't really see the benefit. So Dayoung, who just was recruited, sadly, everyone's leaving my lab. Anybody who knows any students that want to join my lab, I'll be like down to a lab of one, I think, or two in a few months, because they all just got jobs and they're all leaving. So he's going to Virginia Tech as a tenure track professor with a great startup. I'm really proud of him. So what we're trying to do now is, can we get rid of the nanoparticle core and sort of take out the core and just make this bispecific multivalent compounds just purely bispecific. And I think multivalency is very important, but it's not critically important. And so we're looking at ways to create these bispecific engagers, if you will, where on the one hand, it engages with myeloid cells. And these are the different types of tumors that we are currently working on in the laboratory. So this is just a summary that we were able to somehow fool our myeloid cells that are circulating to think that solid tumors are actually liquid tumors, in particular, and therapies that are associated with innate immune checkpoints such as CD47 blockade. I think that may be an interesting strategy that we can move forward with, and just to show that engineering principles, where if you know how to sort of plug and play different peptides together, it's a really interesting sort of area to study, if anyone's interested. So I want to just pivot now in the last sort of 20 minutes or so and talk about a really exciting platform that we're developing. And in fact, we're hoping that this will be translated into a patient or two in the next 2 years, and we're rapidly developing this technology. Unfortunately, not for brain tumors. We had to choose a super rare, even rarer than GBM, a renal medullary carcinoma that has a particular gene that's apparently lost. And if you can replete that gene, the cancer cells do not grow. And so because of that one factoid, and there's one particular renowned clinician whose entire clinical practice is on renal medullary carcinoma. And so we're going to be pursuing that. And so I'm learning about kidney cancers next year a little bit, which is kind of strange. But recently there's been a lot of interest in developing mRNA therapeutics, including Nobel prizes that are given out. And we've all probably received the mRNA vaccine at this point. Not all, but I'm sure most of you guys. And the way that, I'm not going to go into detail here, but just know that core of all mRNA vaccines or COVID vaccines that we have at the moment is lipid nanoparticles. These are foreign materials, as I mentioned. They're spherical nanomaterials that are foreign. They're then surface modified, pegylated, such that you don't interact with the host so much. But the way in which that this is given, you basically allow the mRNA to be introduced to the cell, and hoping that the cell would then produce the proteins. The problem with the mRNA technology that we have right now are manifold. One, some patients really react to the lipid nanoparticles, whether it be just local immune reaction or anaphylactic reaction. There's a lot of non-specific uptake. And there's been studied, depending on who you read, over 95% or more of the nanoparticles that you inject, if you were to inject intravenously, will get all stuck in the liver. And so unless you're trying to cure liver cancer or something to do with the liver, this technology that we have, which majority of the field is utilizing lipid nanoparticles, including Pfizer and Moderna and whatnot, are stuck in the liver. And with that, if 5% are somehow able to bypass it, they really cannot get into the brain or any of these biological barriers because you don't have enough, first of all, but also just the chemistry of it, it has to be negatively charged and all of this, that it's just not bypassing the blood-brain barrier. So we think that we've developed a delivery platform of mRNA that can sort of surpass all of these bottleneck issues. And that's where this extracellular vesicle story comes into play. So extracellular vesicles are little vesicles that sort of come off of every cells in our body. It's the way that your cells communicate with cells that are sort of distant. And so constantly your cells are just like in their homeostatic state and they're sort of releasing these tiny vesicles. And within these vesicles, there are a lot of hallmarks on the surface of it, but also they're carrying messages that sort of are telling other cells that are distant what to do. And so we looked at, and one of the biggest bottleneck in this sort of area is that the ability to create exosomes that are efficient and you can create a lot of it for clinical trial use is a big bottleneck. And I'll show you how we've been able to bypass that with our work. But in the sort of the grand scheme of mRNA therapeutics right now, the biggest sort of space right now is lipid nanoparticles. We've been working on the extracellular vesicles even before the LNP vaccines came out, and so hopefully we'll get that into the clinical realm quickly. And Dayoung and others in our laboratory are looking at different ways to deliver mRNA using peptides. Currently, our lab is focused on cancer, but I will, as a prototype, I'll have to join this story, talk to you about how we can actually deliver collagen for anti-wrinkling system. And that's really been interesting because you have 5 papers curing cancer in papers, but you have 1 paper in anti-wrinkle, and everybody is interested in anti-wrinkle stuff. And so we're hoping that we can maybe, increase our visibility through the cosmetic anti-wrinkle system so that we can maybe feed into the cancer research somehow through that mechanism. And then we have some rare genetic disorders that we're trying to replace, which Kristen is hopefully wrapping up the paper soon and then submitting. So these are some of the direct comparisons between the different gene therapies that are out there. And I've already talked about the lipid nanoparticle and all of the difficulties that we're facing. The thing is that the lipid nanoparticle is FDA approved and we are not. And so we're looking at all of the studies that can sort of enable us to get into the clinical trial and studying FDA enabling studies of different sort of areas. And so these are our technology and the different disease models that we're looking at right now. So this is a story about replacing a protein. And I'm going to start off with replacing collagen before everyone falls asleep, because it's applicable to everyone. And this can be essentially translated to even anti-aging or even rejuvenation, if you will, and things like that. So we as a proof of concept, what we did was we delivered collagen specifically to fibroblasts in your skin, and it really de-wrinkled these animals. And so just as a quick study here, we've developed this platform or this system about 4 or 5 years ago, in which we have, with our collaborators, we have these tiny little microchips. And the microchips under the microscope, you can see tiny holes in them, these nano channels. And why that's important is that when you actually have these cells, these source cells, as we call them, and in this study and some of our other ones, we use mouse embryonic fibroblasts that we grow as a single layer. And this whole system has a pool of sort of fluid that also houses the plasmid of interest. And so we take human plasmid of, but for example, collagen or collagen 1, and that's just sort of like just floating around in the bottom chamber here. And what happens is that when we optimize sort of the electrical force, where we don't kill off these cells, but just gently stimulate them just enough that these nanochannels at the point of contact allows plasma membranes to translate open. That allows plasmid to sort of enter these embryonic fibroblasts or store cells. And also the electrical force allows a significant amount, many folds of these extracellular vesicles to be formed because the cells are under stress and they're trying to sort of send out messages that, something's happening, come and help us. And so what we do is we take the supernatant now and we isolate all of these exosomes that have mRNA for collagen. And so they're all sort of collected. And then what we do is we inject it into the system of interest. And here we have fibroblasts in the skin. And so this is a study. I should actually put your name in there. You're involved in it too, right? Kristen's involved in like every project and more. After she left, I was like, oh, I didn't know that I was involved in that project, but sure, I'll help you with it. And so this is a study that just came out. And that is that we took basically nude animals. These are animals that don't have any hair so that we can see their wrinkling effect. And we essentially induced or exposed ultraviolet light for many weeks to the point where their skin's really wrinkly and it's very tough and fibrous. Patients that you see that are like sunbaked for many years, like the skin's really not that great looking. And so you can see here is that when we inject these exosomes now that have collagen mRNA in them, we inject them, and around about 20 to 30 days over time, the skin like de-wrinkles as compared to some of our other controls here, just saline being injected. And then you have the retinoic acid and some of the other controls that people use in the cosmetic world. When we look at the total wrinkle number, significantly reduced compared to all of our other controls. And so what we did was, can we, instead of just injecting it locally with a needle, can we actually give the same material over a wider surface, much more like a patch? And so we created this biodegradable, biocompatible sort of PDMS system. It's really not novel. It's been around for many like decades, I assume. And we placed tiny little sort of needles, nano needles, if you will. And in those needles, we have these exosomes that are embedded in it. And we can actually alter the PDMS in such a way that depending on the need, so for the skin, we'd like for it to dissolve completely within about 10-15 minutes. Maybe for cancer therapy, you want it dissolving over about an hour or so. We essentially allow it and design it in such a way that the needle tip degrade over time. We place a patch on these animals that are completely wrinkled up. And what we see is that we have on the bottom row here, you see this complete de-wrinkling effect around 30 to up to about 70, 80 days, which we're really happy to see. And you can see all of our other controls like saline, they just look the same, unchanged, obviously. And just needle injection, you do see some improvement, but not as widespread. And that's just showing the difference between before and after. And what was really interesting also is that this patch allow us to deliver collagen not just on the surface, but actually it can be delivered in the subcutaneous area too. And so you can see collagen exosomes. I don't know if you can see these little particles here, blue, that are seen beyond just the epidermal layer. And we did all of these other studies to show that the elasticity, the buoyancy, like hydration, all of those things just improve. And so I just wanted to show you here where if you just do a single injection of this EB that's been now colored in the red, very local focal, and then, yes, you will have some de-wrinkling probably around this area, this square area here. But when you do this patch, you can actually have this system deliver collagen much deeper. And so this is what it looks like in real life. This is just a commercially available applicator that we just used. Apply this on the animal who's been photo aged. It's degraded now. We just wait. And yeah, so that's what it looks like. What's really interesting is that the people that are really into this are like Unilever, L'Oreal, all these, they're looking at the next biologics that go beyond just the cream base, because we know, or they think that the next 5-10 years will be a lot of treatment where the patient will actually come into clinic and see a dermatologist or a plastic surgeon and we'll get therapy. That's their market now. And what this therapy apparently allows them to do is target areas in the body that maybe Botox cannot reach, such as the neck or the hand, where the muscles themselves are so small that you can't physically inject the Botox. So we were able to show in this paper significant large quantities of EVs to be produced that can, an area that we're looking at now is just scale up production for human use. And how does that, look like in a GMP facility? And we were able to show that there's wrinkling of the skin and for protein therapy. And so for GBM, this was our first study in 2019 with Zhao Gang. Basically, what we do is that we have to modify the source cells in such a way that they're able to target GBM cells specifically. So here we use EGFR variant 3, which is overexpressed in certain GBMs, portion of GBMs. And these exosomes now have EGFR variant 3 on the surface. So when you inject it IV, it'll sort of home to the brain that has that marker. I can see that there's a significant increase in brain penetration here when you inject in the tail vein. We also show that compared to all of the other controls, whether if you're targeting, there's significant less with our exosomes, but if we have the targeted one where we have EGFR variant 3 on the surface now, there's significant less of this material stuck in the liver as compared to all of our other controls. And this is what the tumor, the survival and the tumor sort of growth look like with the exosome that's targeted. And we also show, and I forgot to mention, what are these exosomes carrying? So we were able to target, but we were able to put in tumor suppressor genes that are lost or are not functioning. And so here we have P10, but we also can do combinatorial therapy where we can deliver P10 and P53 and replete them. You can see here in this slide where cells that do not have P10 are repleted here with our therapy as compared to the controls. You can see in the Western plot here that the P10 is repleted with our treatment. The last study that I wanted to sort of share in the next 5 minutes or so is our version 2. And again, Kristen, you should be on there because you're on this paper, but I will change it for the next time. She's on all of our papers because she's been so productive. We couldn't get her out of the lab. She was like living there like 20 hours a day. In fact, the campus police during COVID was contacting our lab saying, there is somebody in your lab that keeps coming in during off hours. That's how she was so productive. Anyhow, this is our version 2 that we're using going forward in terms of gene replacement. And that is that instead of having a stationary source cell that's on top of a microchip, we now have a microfluidic device. It's a tiny little sort of fluidic device where the cells essentially flow through this channel. And as they're flowing slowly, electrical energy is being shot through, allowing a significant amount of exosomes to be formed, and you collect them and essentially inject them into the animals. What's also interesting about this version 2 platform is that we were able to surface modify these exosomes, or the source cells, if you will, with CD64. This is an actor marker that you can pretty much mix and match almost any monoclonal antibody, and it'll stick to it. And so if you're interested in, for example, EGFR variant 3 or EGFR or HER2, any antibody that you want to conjugate now, you don't have to change the entire source cell for that particular antibody and change in and out. All you have to do is just mix them together and the antibody will just stick to the CD64. So it's really a plug and play approach. If you have an antibody that targets a cell of interest, whether it be cancer or any other diseases, if you will, you'll be able to target it. We also have markers such as anti-PD-L1 checkpoint blockade and CD71, a way to sort of target the transferrin targeting pathway that allows it to get to the brain, that allows us to utilize this exosome for targeting purposes. And so this is just to show that The system, again, is able to penetrate and enter brain tissue or brain tumor, as you can see here, as compared to some of their other controls. We still see a lot in the liver, but significantly less than the others. What does this look like? And so we took one of the more aggressive syngeneic models of quote unquote glioblastoma, the SB28 system. One of the ways in which people think that this cell line is so aggressive and really poorly immunogenic is that they have very low MHC1. So as I mentioned, that's sort of the molecule that allows antigen presentation pathway to occur with T cells sort of locking. So if you don't have a lot of these, then you assume that the immune cells cannot really work. So we intentionally took these models, and we see that despite the fact that a lot of them should be dying at, about a month out, we're able to improve their survival. And so the question is, can we make this a flat line by doing combinatorial therapy? And that's something that we haven't done yet. And these are some of the MRI images that Kristen, you took for us. And you see the tumors sort of shrinking over time. Well, not over time. These are different controls, but we do see them reducing over time. So this CNP, or we're calling it central cellular nanoporation technology, allows us to sort of bypass a major bottleneck in the EV world, which is producing large quantities of EVs, and it also allows us to deliver mRNA. So I should mention that a lot of EVs, you can actually insert and deliver siRNA or smaller sort of gene products in there. That's not difficult. It's more of the bigger sort of transcripts, mRNA of larger materials. How do you stuff this massive amounts of genetic material into, vesicles that are about 100 nanometers? And so what we do here in this technology, it wasn't so clear, I think I skipped that part, where the exosomes, what they do is that they're carrying the mRNA, they dock and enter the target cell of interest. And when they do that, within about 8 to 12 hours, as the mRNA is now fused and into the cytoplasm, the cell, whether it be the GBM cell or the fibroblast, they start transcribing the mRNA into the protein that we've sort of designed the system to be introduced. So if that is essentially P53 or P10 or collagen that's sort of lacking, we're able to do that by hijacking your body's own cellular machinery that's already, ready and able to do so. And I think that's something that's very novel about this is that we are now, sort of pivoting into the rare genetic disorders where there are proteins that are so large that we cannot physically introduce them into the human body. And by using this technology where you're using the own body to make these proteins, that we're able to replete some of these genes that are missing. And so I think that's like an area that's really, really exciting. And the fact that unlike it is gene therapy, but unlike the classical, changing your genome, where you're, changing the DNA structure, we're actually doing that, changing or altering the amount beyond the DNA. And so we're not really altering your genetic makeup, and it's not a permanent effect. And so I don't know if you noticed that about 90 days for collagen, the amount of proteins that you generate degrades over time. Which is a good and a bad thing. Good thing in the sense that your genetic makeup is not being altered permanently. Bad in the sense that if you're a cancer patient or a patient, you need to have multiple dosing, every 3 to 4 months, depending on the optimization of the system that we're still doing. And so that's something that we're working on as well. How do we lengthen that? But I think because of protein and degradation pathway, we can't really control. We're limited by the fact that every 3 or 4 months, these patients will have to come in for a re-injection. So these are some of the papers that have come out. If you're interested, they're all sort of in the same concept of high throughput, large scale generation of EV and delivering. So here is my laboratory, right? Well, except for Ife right now, and they're all leaving me, sadly, for good reasons, but they all just happen to be leaving at the same time. And hopefully Sungdong has a couple more years. He'll be staying in our laboratory, and some of our collaborators and some of our This is our funding source. I wanted to highlight the ABTA, American Brain Tumor Association, and Uncle Corey Foundation that Hunting was able to get, and others. So she was very productive. And then our laboratory meeting, Athens meeting as well. He's going to medical school. So another student. And that is it. Give you back 5 minutes. Thank you.
All right, thank you so much. What a great talk. If you have any questions, please use, and you're online, please use the Q&A feature located at the bottom of your screen. And while we wait on any questions, I wanted to mention that next week we have Aaron Gillespie from the University of California, San Diego, presenting on implementation science and digital health to promote equitable access to evidence-based radiation oncology. And to view all upcoming Winship Grand Rounds lectures, please visit the Grand Rounds page on the Winship Cancer Center website or the Winship calendar. Any questions? I will check online for us. And if there's any from the room. Oh, we have a lot of chat. Okay, Dr. Kim, you have, well, okay, a couple questions. Looks like. Yeah, I guess we can start from the room first. Yes, Dr. Shu.
The exosomes, they don't really generate immune response. Is that because of the protein makeup of the surface? And does that need to be patient specific? Are there specific factors? Because I would think that sometimes you might get factors that might be more immunogenic if you cross over between individuals.
Right.
Oh, Dr. Kim, if you want to come up to the front, that way everyone can hear, that'd be wonderful.
So we don't see a lot of immune response, and that's the beauty of the system because we're using almost like a syngeneic model, if you will. They're mouse, so mouse and mouse cells don't really react, and so so right now, in order to translate this into patients, I'm learning that to purchase a human, like embryonic fibroblast-like cells are like hundreds of thousands of dollars to just acquire, and there are companies that sell it. So that's the beauty of it, is that your body, think of it as self. And whatever immunogenic material that we're delivering, it's all inside in the form of an mRNA that then go in. So one of the study which I didn't focus or I didn't highlight is the version 2. What we also deliver in that extracellular vesicle is interferon gamma plasmid that gets transcribed into mRNA, and that sort of gets fused into the cancer cell, deliver interferon gamma, and that cancer cell then becomes very pro-inflammatory, and that then triggers all of the immune system. Yeah, but the exosomes themselves do not induce any immune inflammatory. No. We don't think so. What we do need to do for each, not each patient, but each cancer type is we need, so now that we have the adapter marker, we need the targeting agent, so the antibody that can sort of target the EV, and then whatever the content is. Yeah. So we're, we, you don't know that yet, but we're looking at neurodegenerative disorders that have proteins that are altered. We're looking at some of the very rare pediatric rare genetic disorders, and we're right now collaborating with a group from St. Jude's, and then of course cancer. But being that we're at the cancer center, we have, the first patient in trial will go into cancer patient.
How long does the excess mRNA last within the cell? I guess how long the therapy works will somewhat dependent on the half-life protein itself.
Yes.
But I was just curious how long.
So the question is how long do the proteins or the therapy last?
Protein stays.
We think, Yes, and so we did those studies, and depending on the system, I think it was anywhere between 8 to 16 hours. It's around 12 hours where it peaks, where there's a lot of mRNA being sort of introduced. Yes, yeah, so it's all within definitely within 24 hours. I think a lot the peak happened around 12 to 16 hours. So it's not just floating around everywhere.
And we have a couple of questions from Dr. Waller, I think. So he says, which technologies are you developing that can target the immunosuppressive myeloid compartment in GBM?
Yeah, so right now for GBM, what we are targeting is things like, we're not actually targeting any suppressive myeloid cells right now per se. What we're trying to do is how do we make the tumor environment hot by being more pro-inflammatory, and whether that be through cytokines or, in fact, as I mentioned, the interferon gamma, and also different combinations of tumor suppressor genes or mutated genes. And so we're not actually in this, the EV is targeting myeloid cells per se, but we are in our engager sort of model trying to target all of as many of the myeloid cells in the tumor microenvironment itself, and then allowing phagocytosis to occur, hoping that that will turn like a cold sort of tumor microenvironment to a hot one. But I mean, if there is a target, or if there's like a marker that makes myeloid cells less, immunosuppressed, then certainly we can look at that particular gene, deliver the mRNA for it. But we haven't done that at the moment. So another question, it's that SLAMF7 was approved for treating myeloma, doesn't have a single agent activity. So approved in combination with lenalidomide, and can you please describe any work related to its interaction on CD47 animal model? Does this have relevance as priming? Sorry, I got to go to the next slide. Priming for any immune therapy such as PD-1. Yeah, so that's a great question. So for us, the SLAMF7 that we use is not an antibody. It's like a peptide, for one. And number two is that because, as you know, so where we are stuck with this story, and that's why we're looking at different markers of phagocytosis, the CD47 is that Gilead and Innovent and Pfizer have pretty much shut down their CD47 portfolio, given that there's been a lot of bone marrow related adverse reaction, and so they're worried about these adverse reactions that they saw in the liquid tumor patients. And so being able to sort of access the CD47 for human and making it sort of, bispecific has been a challenge at the moment. And so we're looking at different sort of markers and not CD47. So we're not actively pursuing the CD47 as a cascade. This was more of a proof of concept. And yes, I do think that priming, I think anything, it just sounds like you just put in PD-1, PD-L1, just works better. But I think part of it is the resistance mechanism where T cells, that produce interferon gamma is a resistance mechanism by which cancer cells then overexpress some of the immune checkpoints. So I think we haven't tried the combination, but that's certainly something that we could try. But we sort of dropped the CD47 story because of that bottleneck as we were trying to get this into clinical development. Another question, what about molecular? So this is about HER2 and nanotherapeutics, which seem to have better tumor control in the animal models. What about other molecular receptors or ligands, including tyrosine kinase receptor ligands, when you target them with nanoparticle formulation? We have not looked at that as a marker because, well, mainly because of funding, and as you saw how small our lab is, that you got to focus on, an area that, it's a balance. So at the end of the day, why are we using HER2? so multiple models out there. We can sort of do 3 to 5, 6 models and sort of show that this proof of concept, which has never been done before, works. And then so then you can sort of go on to the next step. And also the fact that, an R1 may be able to hire 1.5 postdoc, and that's it. And so we have limited number of sort of models that we're going to, expand until we get to that next stage. But certainly, if anyone's doing this work, tyrosine kinase receptor targeting, and want to collaborate with us and want us to sort of help make the material and to test it out in your system, happy to do that. But that's, it's more of a practical reason as to why we're not using it, not because of the fact that, we don't think that the system will work. Please use the Q&A feature to submit questions for today. Okay, any other questions? Yes, Dr. Lawson.
Do you think we should change the way we invest to try to reduce edema? We would like to avoid steroids, of course, in order that we can give them.
Yes, I think, I'm not an Avastin expert, but seeing what we see in the animals, I think, the premise, and I understand the rationale, you try and starve the cancer cells, and that's why you're giving them massive doses. But I think that what's happening is that it's just temporarily closing the leaking, like reducing the leakiness of these vessels. The MRI looks good for several weeks, and then majority of them just come back with a vengeance, because now what you've done is you have these cells that you've temporarily starved, which is okay. And then once you sort of let go, all of these cells are now like, I think, no evidence for it, no preclinical data, that the cells that sort of have been dormant, now you've really changed their environment like acutely. And so now, they come back with a vengeance. So I don't think, I typically try to avoid, at least in my practice, Avastin as long as possible, unless there's that rare patient where you've done the therapy. I would offer surgery if I can, before I go to Avastin, because we've had several incidences where when they needed surgery, they could not get it for at least 3-4 weeks because of the Avastin. And I know that it doesn't work for cancer. We know that it's not working for cancer control. So it's really just a very expensive, cumbersome therapy that patient has to come in for an infusion. And then they all just, experience a massive, not recurrence, but like a massive sort of edema afterwards, a lot of them. So I haven't personally seen a lot of benefit with Avastin, but I know that, patients who, neuro-oncologists who have a bigger neuro-oncology clinic, with a lot of patients that are seeing this, they probably, at MD Anderson, they probably would place some of these patients on Avastin. I try not to on my patients, so.